Reptiles are a very diverse family, ranging from the legged to the legless, land-born to amphibious, minute to huge. But perhaps the most diverse feature is their methods of reproduction. The way in which they give birth, care for their young, and the mechanisms which determine their sex. Turtles spend most of their lives in the water, but being air-breathing, their young must be born on land. Here, huge numbers of Ridley turtles clamber up the beach to bury their eggs. But it's a chancy business. Many eggs are destroyed as the careless mothers dig up the eggs of others in the attempt to bury their own. Once the eggs are laid, the turtles return to the sea, leaving the eggs to hatch in the warmth of the sand. In due course, thousands of hatchlings emerge to attempt a perilous return to the comparative safety of the sea. The few that survive the ravages of the predators often meet further danger from their own kind. Lumbering mothers returning to bury new clutches of eggs. Crocodilians also need to lay their eggs on land, but the care they lavish on their young greatly contrasts that afforded the young turtles. The mother seeks out a carefully selected nest site to lay her eggs. Then, during incubation, the parents remain close at hand to drive away any potential predators. When the young crocodiles are about to hatch, the mother is drawn to the nest by their chirping from within the egg. With great care, the mother gathers the new hatchlings into her massive jaws. This might look like an untimely end for a baby crocodile, but in reality, she's keeping them safe, ready to carry them on the journey to the nursery pool. Once in the nursery pool, the hatchlings keep close to the protection of their formidable mother until they are large enough to fend for themselves. But perhaps the most marked feature of the diversity of the reptiles is the mechanisms which determine their sex. Many reptiles, including snakes and some lizards, employ the same sex-determining mechanism as humans, genetic chromosomal sex determination. It's no chance that human populations are roughly equally divided between male and female. It is a product of our chromosomal sex determination. For the most part, our sex is determined at the moment of fertilization. Most human cells contain 46 chromosomes, and during mitosis, the type of cell division leading to growth, the chromosomes, having undergone replication, now split longitudinally in half to form two new cells, each with an identical set of 46 chromosomes. These are the chromosomes of a human male just before division in mitosis. Most of the chromosomes form identical pairs, as is seen when the chromosomes are stained to produce bands and arranged in a karyotype. Here, 44 of the chromosomes are grouped into 22 matched pairs according to their shape, size, and the pattern of the stained bands. The unmatched pair of chromosomes are termed the X and Y chromosomes. In the female karyotype, there are no unmatched chromosomes, but 23 pairs, including a pair of X chromosomes. So in humans, a pair of X chromosomes corresponds with female, and X and a Y with male. The Y chromosome contains a genetic switch that switches development away from the female towards the male phenotype. So its transmission to the next generation via the gametes is particularly important. In meiosis, the parent cell divides to give gametes, either sperm or ova. Each gamete contains just one member from each of the 23 pairs of chromosomes. Consequently, while meiosis of female XX cells produces ova, all of which contain one X sex chromosome, meiosis of male XY cells produces two types of sperm, those containing an X chromosome and those with a Y chromosome. So formation of gametes from male cells will produce equal numbers of X-containing sperm and Y-containing sperm. But the meiosis of female cells will produce only X-containing ova. During fertilization, the 23 chromosomes of the sperm combine with the 23 chromosomes of the ovum to give a full set of 23 pairs of chromosomes and a chromosomally determined sex. There are 4 possible ways in which the gametes can combine. So in chromosomal sex determination, the separation of chromosomes during meiosis and their combination during fertilization gives rise to equal numbers of female and male offspring. The sex of birds is determined by a similar mechanism, but in this case, it's the male that has the identical pair of sex chromosomes termed Z chromosomes. The female has unmatched Z and W sex chromosomes. The result of the combination of gametes during fertilization is the same, an equal number of ZZ male and ZW female offspring. The sex of a large number of reptiles is determined by chromosomes, XXXY as in mammals, or ZWZZ as in birds, both mechanisms leading to a 50-50 ratio of males to females. But not all reptiles employ chromosomes to determine the sex of their offspring. Many egg-laying reptiles, such as this loggerhead turtle, rely on a different mechanism, which is dependent on the egg's incubation temperature. And since it doesn't depend on the presence of sex chromosomes, the sex of the offspring isn't predetermined at fertilization. The sex of all crocodilians is determined in this way, and the mechanism of temperature-dependent sex determination has been particularly thoroughly studied in the American alligator. Although the average environmental temperature of a nesting area may be the same, the particular site chosen for the nest and its materials and structure can lead to a spread of incubation temperatures around the environmental mean. American alligator incubation temperatures range from 29 to 34 degrees centigrade. At the lower end of the range, no males are produced. All the offspring are female. At the upper end, they're all male. In between, the male-to-female ratio increases with increasing temperature. Only at about 32 degrees is there an equal population of male and female offspring. But temperature-dependent sex determination doesn't always follow this pattern. With loggerhead turtles, it's the proportion of females that increases with higher temperatures. Males are favoured by lower temperatures. The snapping turtle displays yet another variation. Here, both the highest and lowest temperature extremes result in all female populations. In between, there are more males, but at no temperature is the population completely male. Sexing offspring, particularly at the embryonic stage, isn't always a simple matter. The male and female phenotypes are based on structure, physiology, and behavior. Leopard geckos incubated at high and low temperatures have female gonads. Yet while the low-temperature females are fertile, the high-temperature females are sterile and behave as males. Studies of alligators in the wild and in the laboratory show that incubation temperature affects several other characteristics as well as sex. Newly hatched alligators have light and dark stripes, and higher temperature hatchlings, whether they be male or female, have more light-coloured stripes than low-temperature hatchlings. During incubation, the embryo not only increases in mass, it also grows longer and passes through clearly recognizable stages of development. Higher temperatures induce a greater increase of mass per day, a faster increase in length, and a greater rate of development. This results in the all-male embryos from incubation at 33 degrees developing more quickly and hatching in just 64 days, 8 days earlier than the all-female hatchlings from incubation at 30 degrees. But the incubation temperature doesn't only affect the development of the embryo. Surprisingly, it also influences post-hatching growth, with animals hatching from intermediate incubation temperatures growing fastest of all. Another important observation of alligator behavior is that females seek out nesting sites that will reproduce the temperature of their own incubation. This may be linked with the observation that, in the wild, the alligator population is dominated by females. How does this come about? Starting with equal proportions of males and females and an environmental temperature suited to producing a 50-50 ratio of female and male offspring, we'll assume that the females produce twice their number of offspring that will survive to mate. Incubation at the environmental temperature produces an equal population of male and female offspring. So far, this is exactly the same result as would be achieved by chromosomal sex determination. But an easily achieved variation in incubation temperature of 0.3 degrees will cause a large change in the sex ratio. The lower temperature nest producing a 60-40 ratio, and the higher a 40-60 ratio. So far, the average population over the 3 nesting sites remains 50-50. But as these new females are fertilized and seek nests at their own incubation temperature, the picture changes. The declining number of females seeking high-temperature nests, while producing a higher ratio of males, has much less effect on the overall population than the increasing number of low-temperature females. By the 5th generation, the overall population would have grown a little, but now the sex ratio has changed. From 50-50 to 55-45 female to male. Even if the total population was limited by environmental constraints, succeeding generations would push the overall population's sex ratio closer and closer to the 60-40 ratio of the lower incubation temperature. In the wild, average incubation temperatures tend to be well below 32 degrees, and female domination as high as 8-to-1 is not uncommon. But what is the mechanism of temperature-dependent sex determination? One clue comes from the other characteristics influenced by incubation temperature: pigmentation, rate of development, and post-hatching growth. These suggest that temperature acts on a common source, such as the hypothalamus, or via genes that affect the hypothalamus. Another approach to the problem is to ask, at just what stage in the development of the embryo does temperature have its effect in determining sex? This approach to the problem has been extensively studied in the laboratories of Professor Mark Ferguson at the University of Manchester. The experiments involved incubating eggs at 33 degrees and 30 degrees, temperature extremes which would produce all males or all females. At a variety of days into incubation, the eggs were switched to incubate for just 7 days at the other temperature extreme, then returned to the original temperature for the rest of the incubation period. An uninterrupted incubation at 33 degrees produces only males. When the temperature was switched to 30 during days 7 to 14, 15% females were produced. When the switch was made at later dates, fewer females were produced, until day 49, after which the switch had no effect. At the lower incubation temperature, the effect was more defined. A 7-day switch to the higher temperature only produced males between days 7 and 21. These and other experiments indicate that the sex-determining mechanism takes effect over several days, and clearly, since there are some mixed-sex populations, does not affect all the embryos to the same degree. Similar experiments with turtles show that temperatures cycling about a mean produce just the same results as a steady mean temperature. So the mechanism relies on mean temperature over time, not a heat shock. This stability to short-term temperature fluctuations would be important in the wild, as the temperature of the nest will inevitably change over short periods.
So from our work on alligators, it's clear that the sex-determining mechanism is some kind of integrative event. This contrasts with chromosomal sex determination, where the trigger is at a rather specific time at a specific developmental stage. Now, in temperature-dependent sex determination in alligators, this integrative event may be the dosage of a sex-determining molecule, something that builds up progressively during time until it reaches a critical threshold at a particular stage in development.
This dosage hypothesis explains how you can get a 50-50 sex ratio at 32 degrees centigrade. Individual embryos within the population will vary both in terms of the dose they require and the time they require it. Embryos that require a small dose for a small period of time will achieve it and become males. Embryos that require a larger dose for a longer period of time will not achieve it and become females.
So we're fairly certain that the sex-determining mechanism in alligators involves the dosage of a sex-specific molecule which builds up over development to reach a critical level at a threshold time. Now, one of the nice things about the dosage hypothesis is that it can provide a shared basis for chromosomal and temperature-dependent sex determination. You can get the dose in chromosomal sex determination by putting the gene on a specific chromosome, in temperature-dependent sex determination by regulating the gene by temperature. So the kinds of questions are, what is the nature of the sex-determining molecule? How does it work? What kind of genes encode this molecule? And what sorts of changes are needed in those genes to move from temperature-dependent sex determination to chromosomal sex determination? Because we know that temperature-dependent sex determination occurs in primitive animals, such as reptiles, and chromosomal sex determination in higher animals, such as mammals. Well, how do you go about studying this problem? One way is to look at humans.
Normally, individuals who have XX chromosomes should develop as females, but some don't. Some develop testes and become males. And the reason is that a little piece of the Y chromosome has been added on to the X. And this piece of the Y chromosome contains the male-specific gene. Conversely, there are individuals who have XY sex chromosomes who should develop into males, but don't. They develop into females, because a little bit of the Y chromosome has been lost, and this region also contains the sex-determining gene. So in this way, it is possible to map the region of the Y chromosome which contains the primary male-determining gene, a very small fragment of the Y chromosome.
This has been done. Candidate genes have been isolated. And the most interesting thing is that we have shown that these same genes are present in alligators. So it looks like the genes involved in both temperature-dependent sex determination and chromosomal sex determination are the same. What is different is the way in which the genes are regulated. And this provides a common genetic basis for temperature-dependent sex determination and chromosomal sex determination, something which goes back a very long time in evolution.
So far, we've seen examples of chromosomal and temperature-dependent sex determination in reptiles. But there are other mechanisms. One is parthenogenesis, where males are abandoned altogether. The New Mexico whip-tailed lizard is a hybrid between two species of whip-tailed lizard. The little striped and the western whip-tailed lizard. The cells of the parents each contain 23 pairs of chromosomes, but the hybrids contain a set of both parents' chromosomes, 46 in all. This double set of chromosomes confers female characteristics, and these female hybrids reproduce without fertilization. Consequently, only daughters are produced, clones of the mother.
The finding that the New Mexico whip-tailed lizard produced clones caused quite a stir in the 1950s. But another group of lizards, the Australian Gahara lizard, and this, which is a member of the gecko family of lizards, are causing an equally great stir today. Now, as we've seen, lizards possess a large number of mechanisms of sex determination: temperature-dependent sex determination, chromosomal sex determination, and parthenogenesis. The unexpected finding is that some lizards, such as the Japanese gecko, possess both temperature-dependent sex determination and chromosomal sex determination. These therefore present themselves as intriguing models for sex determination. We know relatively little about them. We do know that there are no hermaphrodites, but we don't know if the animals with chromosomal sex determination can interbreed with the animals with temperature-dependent sex determination. So these gecko lizards are at an evolutionary crossroads. The fact that these lizards possess both chromosomal sex determination and temperature-dependent sex determination makes them an ideal model for studying the evolution of chromosomal sex determination from temperature-dependent sex determination. They are also excellent models for investigating the shared genetic basis between temperature-dependent sex determination and chromosomal sex determination.
Cash.